For a microbe to cause disease, there are 4 basic processes that have to happen. The microbe first has to be able to adhere to the cell of the host. They have to be able to colonize once they get inside. They have to colonize, replicate, out-replicate the host's normal microbes, so they can gain nutrients that is needed. They have to be able to avoid host defenses, and then they have to be able to release toxins and cause infection. So if they don't cause the infection, then they're not going to be able to spread to the next organism. So let's look at these different processes. Adherence occurs when the microbe binds to the cell of the host. And this is typically associated with specialized structures on the microbe's membrane known as fimbriae. These allow the host or the microbe to bind to the host cell. Some microbes can bind to multiple host cells, and some are more specific to different types of cells. Once they bind, then they're going to have to start growing. And so sometimes they'll grow in what is called a biofilm, which we've talked about in the past. Biofilms occur when one microbe binds to a surface and starts replicating, and then other microbes bind to that original group of microbes. This protects the microbes from the immune system, as well as any potential drugs that the host takes. They're also going to have to be able to compete with the host microbiota and tolerate any potential toxins that the host microbiota release. So once they have bound, they're going to start releasing chemicals. This will allow the cell to potentially take in the host cell to take in the microbe. So binding on the surface of a cell is not what microbes typically want. They want to get inside the cell so they can then use that host cell to gain entry into the body, or to protect it from other environments. And so there are different mechanisms of releasing chemicals. Gram-negative bacteria have a specialized secretion system known as an injectosome, which allows the bacteria to release chemicals directly into the cell. This alters the cytoskeletal structure and causes the cell to take in the bacteria. There are different environments where the microbes are going to try and get into the body. So one area is the skin, but the skin is an impenetrable region typically. So if the skin is not damaged, then it's not going to allow penetration of bacteria. But if there's a cut, a scrape on the surface of the skin, or if anything enters, like a needle moves in through the skin, this can allow bacteria to get in. And then, of course, you have those vectors that release pathogens through the skin. And the example I have here is Yersinia pestis with the flea. Other microbes are going to go in through mucous membranes. Mucous membranes line at all the regions of the body that are exposed to the outside. So your mouth, your nasal cavity, your anal cavity, your urethra, your vaginal canal. Those are all openings that have a mucous membrane that surround them. And so this is where most pathogens try to get in, through one of these points of entry. And once they get into the mucus, they then can secrete chemicals that cause cells to take them in. Or if they're in the right environment, they might use our own immune system against us by exploiting our antigen sampling process. So what is direct uptake? So this causes cells that typically are non-phagocytic to engulf them via a process known as endocytosis. Salmonella uses a type 3 secretion system that allows it to cause the host cell membrane to basically ruffle and enclose the bacteria into the cell. And then exploiting of antigen sampling. So in this mechanism, MALT, mucosal associated lymphatic tissue, which I talked about a few slides ago, I think a lecture or so ago, is what samples antigens. And so I told you about how this is how we produce a lot of our immune system. The gut microbes are constantly getting sampled by the MALT. So pathogens use the MALT to actually pull them in. An example for this is Shigella survives phagocytosis by macrophages because it causes the macrophages themselves to undergo apoptosis. It binds to the mucosal epithelial cells and induces the uptake from the cell. And this is how it gets into the system. Some microbes don't leave the cell, they stay within a cell. And so this mechanism would be called hiding. So it basically keeps any of the complement proteins, so nonspecific defenses, phagocytes, as well as specific defenses like antibodies from attacking. And some microbes produce chemicals that stop phagocytes from coming around. So C5a is one of the complement proteins in our body produced by our liver, and it is a chemoattractant to phagocytic cells. So Streptococcus pyogenes produces a C5a peptidase, which degrades the chemoattractant, and that in that way, Streptococcus is able to avoid the encounter altogether. We also have some microbes that live inside the phagocytic cell. Some of these will get into the phagocytic cell via the phagosome, but prior to the lysosome binding, they produce chemicals, pores within the membrane, and then they're able to escape. So this is what Listeria monocytogenes does. This is also what Shigella species do. Some bacteria can produce different proteins that stop the phagosome and lysosome from fusion. So Salmonella species can sense the ingestion process of the macrophage, and as soon as that happens, they start producing proteins that block the fusion process. And some can just survive within a phagolysosome. So they can survive the lysozyme that is secreted into the phagolysosome. And this allows it to actually get prepared and then grow. And that's what Coxiella burnetii does. And then we have some microbes that can avoid recognition. And so, one mechanism for this is having a capsule. So some types of Streptococcus pneumoniae, have a capsule. The capsule actually interferes with the process of opsonization. Opsonization occurs when C3b binds to the bacteria, allowing antibodies to then bind the bacteria, which enhances the process of phagocytosis. Streptococcus pyogenes produces an M protein that inactivates C3b. So again, if it's inactivating C3b, then we're stopping that opsonization process. And lastly, some bacteria, both Streptococcus pyogenes and Staphylococcus aureus, have receptors for the Fc region of antibodies. So antibodies have the binding region, which is up here. This is where the antibody binding region, this is where they bind normally to a pathogen and allow for opsonization. But these Fc receptors bind this region of the antibody. And in this mechanism, the pathogen is no longer going to be opsonized and therefore will not be phagocytized. So pretty awesome little mechanisms these microbes have for evading our own host defense. I am going to stop here and we'll get into the damage that is caused in the next video. Bye.